Karen Macaulay. I'm the Clinical Alliances Manager for REPROCELL Europe, and I'm based here in our Glasgow site. So I'm going to kick off the proceedings today, and we'll be chatting to you a bit about the REPROCELL Human Tissue Network for drug discovery. And with particular focus today on why we need fresh human tissue assays for preclinical drug discovery, what the benefits and the challenges are of procuring tissue, and then a little bit about the actual network, how we operate, and then finally to drill down to discuss accessing fresh skin tissue in more detail. Then hopefully, technology permitting, I will hand over to Graham, who's going to talk a bit more detail about the assays that use the skin tissue that we obtain via the network and how these may be useful for your drug development purposes. So why do we need human tissue models to aid drug development? Well, I'm sure that many of you will agree that there is an extremely high failure rate across the drug development pathway. Of the drugs that enter clinical trials in humans, over 90% will fail. And most of this failure occurs in the sort of early stage trials. This situation has not changed much over the past 10 years or so. As you can see from this recent analysis in Nature Reviews, the 7% success rate for drugs entering phase 1 and then reaching the market has remained pretty constant in that time. So investigation of why there is such poor translation or success rate has identified that the main areas of failure are around safety and efficacy. And together these account for around 70% of all failures that we see. And additionally, once drugs actually reach the market, the data suggests that efficacy can be poor at the population level, with greater than sort of 70% of patients receiving no benefit for particular medications that are on the market. So there is clearly a failure to get effective and safe compounds to the market. So why is this the case, or why is there a lack of this translation from preclinical drug development through to clinical trials and ultimately use in the population? There are two main schools of thought as to why this is the case. The first is that human clinical trial design and implementation is perhaps not as optimal as it could be. So areas such as outcome selection, eligibility criteria, patient recruitment, etc. could all be improved or standardised better. The second school of thought is that there's an over-reliance on animal studies in the preclinical stages. There are two competing perspectives here. The first, that animal studies are on the whole useful in terms of general predictability, and that it's more to do with poor model selection and design, along with sort of the reproducibility that are the problem. So similar to the sort of clinical design, trial design issues. The second perspective is that animal to human predictability is poor due to issues around more sort of complex biological systems and species differences in terms of physiology, genetics, etc. And that these do not extrapolate well to the human situation. So both schools of thought were highlighted in a failed trial in the UK back in 2006. If you can cast your mind back that far, this was a phase 1 trial for the CD28 agonist, TGN-1412. And this was a double-blind, randomised, placebo-controlled trial to establish safe human dose for subsequent trials. It had extensive preclinical evaluation, as you would expect, both in vitro studies for cross-reactivity, receptor binding affinity, etc., and an in vivo model, which for this case they used the non-human primate model. This was chosen because of similar receptor binding affinity, receptor homology, and conservation of the various signal transduction pathways to that of humans. And a dose study in the animal model showed that the no observed adverse effect level for this compound was around 50 mg per kilogram. In the actual trial itself, they used the minimal anticipated biological effect level, which was 500 times less than that believed to be the safe dose based on the animal data. And all 6 volunteers were injected, all became ill pretty quickly after injection, and all ended up in intensive care with multiple organ failure. Unfortunately, in this case, there were no fatalities. However, subsequent review and evaluation of this trial highlighted a number of areas where improvement could and should be made. This included the interpretation of the animal studies themselves, insufficient human data, prediction of the dose based on animal models was thought to be unreliable. The donor interval in between injections was considered inadequate, and there was a lack of just general preparedness for the unexpected. And the points highlighted here cover both the clinical trial design and the pure animal to human translation viewpoints. But from a human tissue perspective, points 2 and 3 are of particular interest. And it raises the question, would a good preclinical human tissue-based model have helped in predicting the observed adverse responses in this case? And as a result of this study, various aspects of clinical trial design have been changed, such as the interval between dosing volunteers, etc. However, even with such changes, a more recent study in 2016 had similar drug safety issues at the phase 1 trial of fatty acid amide hydrolase, BIA 102474. Again, as with the previous study, extensive preclinical animal studies had been performed with 4 different species used. The single dose administration arm of the trial was completed with no adverse effects observed, but the multi-dose section of the study resulted in 6 hospitalizations with patients had stroke-like symptoms and 1 actual fatality, which was very unlucky. The review for this particular study is still not complete, and certain information has not been released, but the dosing regime was based on the animal model data, and no investigation of off-target effects in animal or human tissue was performed. So again, would the addition of human tissue testing have helped avoid this fatal failure of a phase 1 trial? So for any model to be valid and useful in predicting human outcomes, there are 3 sort of basic criteria that need to be addressed. The first criterion is model-centric. Are the endpoints similar? The second is pharmacology-centric. How well does the model actually replicate human pharmacology in terms of absorption, metabolism, etc. And thirdly, population-centric. Does the model replicate human diversity enough? And animal models may fail on one or several of these points, and this may account for why there is poor translation to humans. So what is the solution? Well, the solution, one that we believe, is for more human tissue testing in the preclinical drug development processes. And I just want to quickly show here a couple of examples from the data we've produced in the lab demonstrating different responses between animal and human models. The methodology is similar in both examples here. Subcutaneous arteries from both human and dog skin were dissected and used in a contractility model. And the responses in terms of contraction and relaxation of the vessels were measured following the addition of a serotonin agonist on the left and a PDE2 inhibitor on the right. In both cases, there was clear difference in the responses that were observed, with the canine model failing to predict the human response effectively. This kind of data provides a checkpoint in the preclinical development process and shows that much can actually be learned by using human data either on its own or in combination with an animal model. So what are the benefits of using human tissue in preclinical testing? Well, firstly, there's no molecular mismatch due to genetic species variation to consider. Biologically relevant systems can be observed, endpoints are directly comparable, and genetic diversity can be incorporated. And human data can be used to help validate the animal models that we do use, as we saw in the previous slide, making sure that there are no surprises later in the process. In a recent survey of toxicology, international toxicology and safety pharmacology community published a couple of years ago now, and participants were asked to consider what they thought was the main drivers for human tissue use. And unsurprisingly, relevance to the responses seen in humans and poor predictability or a lack of a good animal model were the main considerations. Benefits also includes the ability to use human tissue at all stages of the drug development pathway, from target identification through to lead optimization and preclinical safety. So this, using human tissue at all levels, provides a sort of level of consistency that helps to de-risk the whole process, which is obviously an advantage. There is also clear benefit to personalised or stratified treatment approaches. As the medical field moves more towards this direction, the need for good human tissue assays will only increase. And again, I've just shown an example here from our lab, showing patient responses in response to 2 different drugs, formoterol on the left and salmeterol on the right. The model is measuring bronchodilation in isolated human airways. And what we see here is that the formoterol, we see no patient differences, but with salmeterol we do, and that these differences actually correlated with phenotype. So the human tissue assays can help to differentiate these differences early in the development process, and ultimately could improve the design of subsequent clinical trials and target populations better. There are also economic and ethical benefits. By using human tissue assays, we can meet the rising demand for non-animal alternatives, and any sort of moves by regulators to include human data in future approval processes. And from a financial perspective, preclinical human data can act as a checkpoint to development, helping to reduce further unnecessary costs by stopping a development of a drug earlier in the process if the effects are not seen in human tissue data. The converse is also true, in that drugs may fail based on animal data, but actually look good based on the human data, and so potential drugs could actually be taken through the process. So there are many benefits to using fresh human tissue, but there are obviously certain challenges as well. Accessibility is probably the main challenge for most researchers. So the availability and supply of a particular tissue type, the stability of the tissue, the availability of clinical data, the actual infrastructure, is it there to do the collection, and obviously the ethical approvals need to be in place, and all these factors can impact on accessibility. Variability is also an issue. As scientists, so we like things to be reproducible with little or no variation, to make all the analysis at the end easier. And in terms of donor variability, this can obviously be an issue. However, as I've mentioned just previously, this could also be considered a benefit when we consider the population-centric validity of a model. And thirdly, acceptability. Obviously, regulatory bodies still require testing in animal models as a measure of safety, and there is this perception that human data is perhaps less well validated when compared to animal data. And again, in this recent survey, participants were asked to consider the barriers to human tissue. And again, unsurprisingly, ethics, tissue supply, protocol standardization, validation of models, donor variability, etc., accounted for around 2/3 of the responses received. However, the challenges, are beginning to be addressed. We now have biobanks that have been set up in various institutions to help with tissue collection, and these can be really good uses for obtaining tissue, but there are still differences here in terms of the operational variation between banks. ISO standards for biobanking have recently been introduced also, and regulatory bodies are now looking for human data in some situations. So as these become more embedded in the systems, the challenges may become less. However, even so, to overcome these challenges is time-consuming and a costly effort, and this can hinder many researchers and companies from actually going down this route. So here at REPROCELL, we've tried to address many of these challenges with the REPROCELL Human Tissue Network. And the network consists of 2 areas: a large biorepository and a custom tissue collection for preclinical drug development. So just on the biorepository first, this contains over 600,000 clinical samples from healthy and diseased categories, and is part of a larger network capable of providing global access to samples. All the samples that are collected are collected via informed consent processes and carry extensive donor information sets. The repository contains and has access to frozen tissue, FFPE blocks, DNA, RNA, serum, plasma, etc., and is a great resource for target identification studies. And within the bank, in the biorepository, we have a number of samples from dermatological conditions such as psoriasis and atopic that can be used for research studies. So moving on to the Fresh Human Tissue Network for preclinical drug development. This was established in 2002, initially as a local custom collection here in Glasgow. And we now collect from sites in the UK, US and Europe, and have tissue supply agreements in place with a number of biorepositories, clinicians, surgeons, transplant networks and other commercial partners. And the network has approval as a research tissue bank in both the UK and the US to collect from all of these sources and for all tissue types. Our laboratories are strategically placed to interact with all of our clinical collection sites. So we have one here in Glasgow and the other is based at our site in Maryland in the US. And both sites can operate 24 hours a day, 7 days a week, and coordinate with our supply sites to implement standardised collection and shipping protocols for all of our tissue types. We receive fresh tissue from 3 main collection routes: non-transplantable organs from the transplant networks, tissue that's residual to surgery and diagnostic requirements from hospital sites, and dedicated research sampling. We can collect normal and diseased tissue, and also we can access demographic and some clinical data. This slide is just a snapshot really of some of the tissues we routinely access for projects. And more information on the different tissue types that you can access are available on our website if you're interested. So the network has been in the making now for nearly 20 years and has been specifically developed to try and address many of the challenges to accessing good quality human tissue for research. It's taken quite a bit of time and a lot of effort to set up, but concerns about ethics, standardisation of collections, supply and quality of tissue, access to clinical information, etc., have all been addressed by the network and systems implemented to coordinate these various aspects. So when utilised in conjunction with our validated assays, some of which Graham will discuss shortly, it offers a streamlined process, acting a bit like a human tissue research department for drug discovery, and alleviates many of the sort of practical and perceived barriers that researchers may have, and ultimately, reduces the costs to this type of research. So for my final few slides here, I just want to focus on access to fresh human skin tissue in particular for preclinical drug development purposes. Skin tissue can be collected from all 3 of our supply routes, so surgery, transplant and dedicated sampling. We choose the best route depending on the assay type or the questions to be addressed, the cohort criteria, the sample or endpoint criteria, and of course on any ethical considerations. We then ensure that the protocols meet the individual study requirements and develop these accordingly with various clinical sites. So for skin tissue residual to surgery or via the transplant network, we can obtain quite substantial pieces of tissue with or without the attached subcutaneous fat layer. Tissue sections are full thickness and usually from healthy individuals. And we collect into a suitable transport medium and arrange to ship to the lab within 1 to 6 hours, dependent on the site location. We have regular access dependent on surgery schedules, and our 24-hour sort of 7-day-a-week lab service allows us to capture the majority of surgeries and transplants that are occurring in our sites. And we can access clinical information from these donors through the clinical sites also. Some donor information can be applied to these collections depending on the needs of an individual project, so demographic restrictions or medication restrictions, inclusions, etc. can be applied. And from this tissue we can collect resistance arteries for use in vascular studies or prepare multiple full thickness biopsies. These can be of various size, dependent again on the individual assay or project, and these are usually used in our inflammatory organocultural models. For dedicated research studies, we undertake biopsy sampling. Donors are recruited via patient clinics or clinical trial units on an individual study basis. And as with the surgical route, donor criteria and screening can be applied. So in addition to demographic and medication restrictions, we can also recruit based on a particular disease or disease severity. So for example, atopic dermatitis donors with a particular EASI score. Ethical restrictions do limit the number and size of any samples collected via this route. In general, we can obtain 3 biopsies per patient in total. These can be lesional or non-lesional, and generally are around 3 millimeter size in terms of the punch biopsy. And because this is a dedicated sampling process, we also have the capability to perform additional sampling for blood or urine, and we can also take photographs of lesions if that's something that's required. So our skin tissue access has been developed to try and address as many different research areas as possible, with certain collections being better suited than others to answer specific research questions and use of different assays. And Graham will touch a bit on that in the next section of the webinar. So I just want to finish with a slide highlighting a summary of the various projects that have been facilitated by the REPROCELL Tissue Network over the past 3 years and focusing on skin tissue. So on the left, we have the number of donors and the number of projects utilising skin tissue in red and greens respectively. And on the right, we have the projects by type of assays, so either vascular or organoculture. So far we've procured around greater than 250 donors for skin tissue and have run 45 individual projects, 19 of which have been for our vascular models and 26 which have been for our inflammatory dermatological organoculture models. So the network has been and is an extremely useful component to our preclinical skin-based research options. And from my perspective as the clinical alliances manager, this is great to see that we can access these samples and they can be used productively. So I'm going to hand over now to Graham, who's going to talk a bit more about the different assays and how the skin tissue is used in these assays. So Graham, over to you.
Okay, that's great. Thanks, Karen. Hi, everyone. As Karen said, my name is Graham McCluskey and I head up the R&D team at REPROCELL Europe. I'm also based in our Glasgow lab. So, again, really the main focus of the following slides that I'll walk us through are really, I guess, just to talk through the practicalities of what you can do with these types of skin samples in the lab. I guess the short answer to that is a lot, but what I will do over the slides is just try to show some a pretty top level information about some of the assays that we use in our own lab, just to give you an indication of the types of applications that they can have for you. Okay, so I guess the the features and benefits of using these types of systems in your lab are, the obvious ones are really the clinical and disease relevance. They're from human beings. If they are disease biopsies, then they're from human beings with skin diseases, so that the relevance is quite obvious, really. It obviously has limitations being an in vitro system rather than in vivo, but but for in a study of local skin biology, there's not really anything else that can compare to it. The actual skin biopsies themselves are actually are really flexible. They have a lot of different cell types in there, so there's a lot of different pathways that can be activated, that can be modelled. If you want to look at biology or to look at the modulation of those with test articles. So again, I can give some examples of that. And then actually the throughput for a human system is actually quite good, especially for the normal skin models which come via the surgical route, where we can actually obtain quite large samples. Okay, and again, I can give examples of that later on. And then, as with most of these types of experiments, the list of endpoints are really almost endless. You can look at protein release. You can look at gene expression transcriptomics, some proteomics, immunohistochemistry, and then also, because it's a human system, then we can also look at the actual tissue itself and actually look at the genomic profile of the patient, which can actually be really important if you look to want to marry up that relationship between the genomic background of the sample and also the functionality of it. Okay, so firstly, how long is it possible to maintain skin for? And in the lab, and for the culture experiments and the methodology that we use, it's really up to approximately 1 week. Again, that depends exactly on the media change regime and the size of the biopsies. But there are some changes over that week. You know, as like a time goes on, the the kind of cells and the epithelium, it do change slightly. So so the vast majority of our experiments are actually over that initial kind of 4-day window. Okay, so around about kind of 48 to 72 hours is really the sort of, I guess, the optimal window for most assays. There are some other assays where the endpoint, it is possible to go a little bit further, and really that's just part of the kind of validation of the model to determine that. Don't know what kind of lasts in culture, but how do we do that? And again, there are kind of various different methodologies out there. They're all very much the variations on one theme, and that theme being that you maintain the skin at the air-liquid interface in culture, so the epidermis is exposed to air as it is in the body, and then the biopsies feed from the underlying media. So that, as I said, there's various kind of different ways of doing it, but that's really the kind of key points for culturing skin. Okay, when you get to the biopsies, obviously from a patient kind of clinical biopsies, they arrive in the lab as biopsies. For the surgical residual tissues, they will arrive as a larger piece of tissue, where we biopsy that ourselves in the lab, and that gives us a little bit of leeway to maybe make the biopsies of a different size depending on the application of test article, or maybe even the endpoint, how much material we need for that endpoint. As I mentioned before, these are, although they're very small tissues, they're very, very complex. They are like a multicellular. They have a kind of 3-dimensional structure in architecture, and when we really kind of zoom in on that biology, when there's lots of different pathways which are relevant to disease and cell types which are relevant to disease, in particular inflammatory skin diseases, which can be utilised in these models. In the biopsies from kind of clinical patients, you will have inflammatory infiltrates present within the skin biopsy, but even for the a normal healthy skin samples we receive, there are a skin resident inflammatory cells which can be activated, for example, in order to activate specific pathways to investigate. Okay, and I will give examples of that as we kind of move through the slides, just to give you a flavour of it. So firstly, I'm going to just give you an example of the types of things we can do with the punch biopsies from disease patients. So the slides here focus on kind of psoriasis, but again, exactly the same or kind of similar kind of scenario could be performed using another kind of skin disease. It's really dependent on what type of patient cohort or background you wish the clinic to kind of recruit. Obviously, from a kind of biology perspective, these really are the kind of gold standard for kind of local skin biology of disease. When you do have kind of limited scale as far as the number of biopsies we can obtain from a patient, but it is quite easy to scale to a high number of donors. When the clinics kind of generally, as long as the stipulations on patient criteria are not too high, then when the clinics do find it generally quite easy to obtain patients to come in and provide samples to donate. It gives you the opportunity to look at kind of lesional and also kind of non-lesional skin from the same donor. And again, as are all the skin models, they're very flexible as to what you can model and also what endpoints you can utilise. And again, they're also suitable for ex vivo culture, so we can culture them for a period of time. The phenotype is maintained, and I've got some data on that later. And that also allows us as well to add some kind of drug treatment into the culture as well, so we can look at the ability of a drug treatment to modify a biological process. And I guess a kind of typical kind of study design for that, when we were to take them out to culture, would be really a kind of 24-hour or 48-hour experiment where we're taking the samples from the clinical site and then we're placing them in the culture system that I explained earlier on, and then at the end of the culture period we're harvesting, for example, again this is just an example, when we're taking the supernatants and measuring the kind of protein release, so kind of cytokine chemokine release by ELISA, and then we've taken the biopsies at the end of the experiment. We're going to stabilise them in something like RNA later, and then we've done some kind of gene expression analysis on it. Okay, so what you get from that at the end is something like this. I say this is an example where we've looked at the different biologies of the non-lesional and kind of lesional biopsies between patients. This is a mean data across kind of 4 donors. As you can see, the kind of gene expression profile is very different between both types of biopsy, as you would expect, and that is also maintained in culture. Although it's just a non-plaque versus plaque here, it could easily be a drug treated versus non-drug treated. Okay, so it just gives you an example of the type of assay that you can use for these tissues. And then I'm now going to move on to, the application of the kind of surgical, the kind of normal skin tissues. So these are a lot larger, which gives us a little bit more scope, but the general principle is exactly the same. As I said previously, there are resident immune cells within these normal skin samples, and you can activate pathways of interest by adding artificial stimulants to the system. So we have a range of different stimulants we use depending upon pathways we want to mimic. Here I'm going to present some data on an atopic dermatitis cocktail which we've developed in-house, which really kind of targets on kind of stimulating the Th2 pathways, which is obviously relevant to AD. And when we add that a cocktail to in the culture, we see a change in phenotype, as you would expect. So on the left here is a volcano plot of a sort of wide array of kind of gene expression that kind of shows the differences between the non-stimulated group and then the a stimulated group with our cocktail. And as you can see, there's there's a marked change in a phenotype there. You have some kind of defensins upregulated. And then if we kind of focus in on the Th2 a profile in particular, then that's on the right hand side when we're looking here at the kind of gene expression changes that happen to IL-13 gene expression and also IL-5, which are really those a key and kind of hallmark changes you would expect by activation of those Th2 pathways. Here it's just kind of gene expression, but we also see the same trend as well with kind of protein release. So we see the subsequent increase in the release of the cytokines themselves. So we see an upregulation of IL-13, which is the left-hand graph. And again, this is a non-stimulated versus cocktail-stimulated groups. And then IL-5 in the middle. And then we also do see an increase, although it's quite low levels, we do see an increase in IL-22 as well with the cocktail. Okay, so the fact that we have this kind of stimulation window in the system of of an AD a relevant a pathway, then that allows us to test the ability of a novel test compounds to modulate and also inhibit that stimulation. So here is just some example data again where we have tested some drugs of interest which are obviously of kind of clinical relevance. The mechanism is of a clinical relevance to atopic dermatitis. We have a steroid here, a betamethasone, which is the red bars. We have the green, which is a kind of JAK inhibitor, and then we have the purple as a kind of PDE4 inhibitor. When the graphs at the top left and right are just kind of showing the a cytokine release from the biopsies. The control group is kind of stimulated but non-drug treated, and then the subsequent bars are the kind of drug treated. So you can see there's a kind of significant inhibition of a cytokine release in those treated biopsies. Now here we've just looked at a very high concentration of each of the drugs, but as I said previously, because we have a larger scope of number of biopsies obtainable from the healthy surgical samples, then we have enough biopsies to allow us to measure a concentration dependent effect to drugs as well. So again, this is just some example data taking betamethasone as an example here, where we've looked at the concentration response in the assay as well, and as an endpoint we're just measuring IL-5 release into the culture media, and that allows us to calculate things like EC50 values. Again, because it's the normal skin and healthy skin model, then we can actually use larger size biopsies, and that allows us to apply compounds and formulations topically as well to the model. All the previous data that I've shown you was applying a test compound to the culture media, which is really a little bit like a systemic administration, where here we're applying them as a topical formulation to the stratum corneum and the biopsy itself. So again, it's exactly the same result when we apply topical compounds. We can measure the anti-inflammatory or inhibitory effects of these compounds on the stimulation profile. Okay, so that gives you like an idea of the types of things you can do with the fresh skin assays themselves. What I'm going to do now is I'm going to move on to like a little bit more kind of R&D and a little bit like more like a cell culture work. It is possible to take the human skin samples and to isolate a primary cells. As I said, one of the examples of the human skin models that I've just described, and one of the advantages is the complexity of them is a kind of real advantage, which makes them really flexible to measure a multiple different things, and also makes them adaptable to different kind of mechanisms and pathways. But for some applications, then that can introduce quite a lot of, I would say, kind of background noise to the system, which for certain mechanisms might make it difficult to obtain a good assay window. So in that case, it is possible then to look at a simpler kind of biology, such as kind of 3D, a reconstructed kind of skin models or structures. Although it is simpler, it's still quite complex. It's not your kind of flattened kind of 2D a cell culture. There are a lot of isolation protocols available for different cell types out there. On the picture on the right, that's just a kind of 3D, a reconstructed skin model with two cell types, just like fibroblasts and keratinocytes layered on top. But there are, it's possible to isolate other cell types and build them into different models. The access to different patient profiles, that does give you that ability to compare the function of diseased cells versus non-diseased cells in these types of models, and the models themselves do lend themselves to being slightly higher a throughput than your fresh human skin biopsies. You are able to miniaturize them in size. However, when building these models, it is very important to try to maintain as much as possible, the kind of relevant human cell environment around these cells. We do notice big differences in the function of cells, when they are structured correctly, and also in their own and also kind of native extracellular matrix. And we're actually quite lucky, at REPROCELL, to have our own a 3D scaffold, which is called Alvatex, the one which we use to build a lot of these epithelial structures. That allows us to build these really nice, layered, stratified epidermal models on top of kind of dermis. So I'm going to just talk you through an example of how we've kind of used a kind of 3D a cell system to model an aspect of skin disease, with the main focus being a skin fibrosis. Okay, so what we have been working on is a model where we're taking human primary fibroblasts and then we're kind of seeding them into our Alvatex scaffold. It is just a cell type system, but we are aiming to look at the extracellular matrix deposition within that Alvatex kind of scaffold over time. We're not adding any other types of ECM components. We're allowing the human fibroblasts to deposit on their own ECM, which again we found very, very important in the function of the cells. If you start to put in any other types of, say, kind of collagens, etc., then the fibroblast function does change. Okay, we stimulate that with TGF-beta, and when in doing so, we do see a deposition of collagen fibers within the Alvatex, and then that allows us to firstly to quantify that, but then when the system itself also allows us to add some kind of drug treatments to the system as well to see if we can inhibit that, and also to look at the different kind of profiles and mechanisms by which the drug treatments are having an inhibitory effect. Okay, so this is my last slide, and it's just some example data from that model as well, just to give you a flavour of the types of outputs you can get from these systems. So on the left-hand side, there's some immunohistochemistry, like where we show the ECM, like a deposition within the model itself. So as I said, we're not adding any, exogenous, like ECM components to that. All of the ECM that's kind of generated that we're measuring here has been laid down by the fibroblasts within the scaffold structure that we have used. It is increased, as you would expect, with the addition of TGF-beta. And then on the right-hand side, that allows us to then to quantify the inhibitory effects of some test compounds. Here we've tested 2 test compounds. They have been added at the same time as the TGF-beta, and as you can see, we can see a nice kind of concentration dependent reduction in the amount of a collagen that's been deposited over the time of culture. Okay, so that allows us to look at things like EC50 again, and allows us to characterize the compound response in the system. So that's really the end of my slides. Again, there are kind of lots of different things you could do. I just wanted to give you some examples just to give you a flavour of it. And really then that's us just ready to open up for questions, and kind of thanks again for your time.